So with this video short, I'm going to be talking about and explaining the structure function analysis of collagen. This protein is going to be very different than hemoglobin and myoglobin. In fact, the role of collagen is a structural role. So when we talk about collagen, there are a few things off the bat that are very different than in hemoglobin. So the first thing, collagen is a triple helix. So it is 3 polypeptide chains that wrap around each other. And each chain of polypeptides is roughly about 1,000 to 1,400 residues long. All right, these 3 polypeptide chains wind around each other, kind of like how a rope, you could imagine how a rope does. All right. It has what is referred to as it's called a triple helix, but this is not like the same alpha helices we see when we talk about secondary structure. This is a very different type of helix. It doesn't have almost any of those same properties that the alpha helices have. And furthermore, where is collagen located? So collagen is actually 30% of all proteins in your body that your body makes is collagen. So collagen's in our skin. It's the structural kind of backbone in which our bones and our teeth are built off of. And it also is commonly found in tendons, right? And these tendons are what binds our muscles to our bone. Okay. And humans, there's 28 different types of collagen, but we're just gonna kind of pretend there's just one type of collagen, but the different types are expressed in different areas of your body because they have slightly different primary sequences, and that creates a little bit of diversity between those collagen. Now, they'll still all have that triple helix rope-like structure, but their amino acid side chains will differ just a little bit. Okay. The common recurring pattern of the polypeptide chain is goes by 3s, and it'll go glycine, X, and Y. So we've got X is usually a proline residue, and the Y is usually a hydroxyproline residue. Now, there are other residues. Every now and then there's a lysine, and that comes into play with the strength of our collagen molecule. But again, so I want you to remember collagen has a 3 amino acid-ish repeating pattern. Glycine makes up about 30%. So it's about every 3rd residue is a glycine. Now, that's kind of interesting because glycine is the most flexible of all the amino acids, right? It has no side chain, it's just a proton. So there's not a lot of steric hindrance you'd have to worry about, which is what allows that polypeptide chain to wrap around each other, all 3 of them. So instead of a alpha helix, which normally has about 4 turns per residue, the collagen chain has a lot more. So it's like a corkscrew, but stretched out really far. So glycine's allowing for that flexibility. And what's also, what sounds just crazy is that proline is probably the 2nd most occurring residue, and that's the most geometrically constricted residue. And so the fact that the glycines are present accounts for some of that flexibility in getting that helix to be stretched out so far. But the prolines add a rigidity aspect to the collagen, which is important for its function of being a structural protein. So collagen does get its strength from 2 different major interactions. So the very first one is interchain hydrogen bonding. Okay, so remember we have 3 helices that are wrapping around each other in a left-handed fashion, and the helices themselves are hydrogen bonding with one another. So interchain, like an interstate is a highway that travels between all the states. Interchain hydrogen bonding means that the N terminus of one chain is hydrogen bonding with the C terminus of a separate chain. So it's very different than a normal alpha helix, which in our normal alpha helices, you would have an intra-chain hydrogen bond where, right, the N terminus would bind to the C terminus 4-ish residues away in a normal alpha helix of the same chain. So that's one thing that kind of keeps those 3 helices wound together is the interchain hydrogen bonding, because it's going up the entire thing. And another thing is that we'll see these three triple helice bundles will stack with each other to make fibrils, and those are fibrils, and those fibrils are covalently linked together through some modifications of lysine side chains. Okay, so it requires two different lysine side chains on two different triple helices. One will be modified so that the lysine becomes an allelcine, so it has this aldehyde functionality at the end. And the other one's just a regular lysine residue, and those two will react. A water molecule will leave, and you'll form a Schiff base. And that now covalently tethers two separate triple helices together. Okay, and so you keep building and building and building, and you get those bigger fibrils. Okay, there are a couple other post-translational modifications. So this changing of a lysine to an allicine or allelcine is a post-translational modification. Post-translation means after it's come off the ribosome, so after it's been translated, and a modification essentially it's a chemical reaction that occurs on that amino acid or on that polypeptide chain somewhere. So the two, again, most common post-translational modifications are the formation of 4-hydroxyproline. So you remember it's about 30 every second, or it's the X in the gly-XY repeating pattern. So the X's tend to be prolines, and then the Y's tend to be hydroxyprolines. And so hydroxyproline is just an enzyme that oxidizes proline by adding this alcohol group on the 4th position. And this is the 4th position of the amino acid, because carbon 1 is going to be our C-terminal carboxylic acid carbon. That's our carbon 1. Then you just follow the chain around, right? 1, carbon 2, carbon 3, that's carbon 4. That'd be carbon 5, and then that would be nitrogen. You could also call these by their, that's the N-terminus. You can call some of these residues also, though, by their Greek name. So recall, here's our C-terminal, here's our C-terminus, here's our N-terminus, and this carbon we call the alpha carbon. So this would be the beta carbon. That would be the gamma carbon. So this could also be called gamma hydroxyproline. Yeah, just keep that in mind. A second common post-translational modification, and this has to deal with how you generate that allele lysine or allylysine residue, is 5-hydroxylysine. So again, it's just an enzyme that binds onto some of these lysines and oxidizes them by adding on an alcohol functional group. Okay, this is 5 because remember our carboxylic acid carbon's carbon 1, our C-terminal carbon's carbon 1. So there's 1, 2, 3, 4. It's on the 5th carbon. In order for those oxidations to occur, it goes through, we'll look at proline specifically. So you'll take just a regular old-fashioned proline, and through this enzyme called proline hydroxylase, this is how you create that 4-hydroxyproline. So it adds on that alcohol functional group. Okay, this requires the presence of oxygen and iron center. So we're going to have an iron center, iron 2 center. And then we also have another co-substrate up here, this 2-oxoglutarate. And this kind of comes from the citric acid cycle. That's where that kind of can come from. But so what you'll see is that this oxygen will be the oxygen that gets at one of them, at least will be added on here to form our alcohol. And then after we perform this oxidation, the iron converts to iron 3+. And in order for this reaction to occur again, you're going to have to turn iron 3+ back into iron 2+. And that requires a cofactor, vitamin C. And where I'm getting to with all this long story here is that if we think back and maybe you know a little bit of history about sailors, but the pirates were notorious for having this condition called scurvy. And scurvy is usually kind of diagnosed by bleeding gums, like swollen and bleeding gums, sunken eyes, easily bruised. And this is occurring because the lack of vitamin C, which affects the collagen production. Okay, so here's kind of, like scurvy is not the best. So if you don't have enough of that vitamin C cofactor, your body can't maintain the collagen through kind of the daily knocking and daily use and abuse of your body, right? You're chewing food and you're smashing your gums, and the collagen's also there in the tissues of your gums. And so if the collagen's not able to fix itself after you keep on banging it up and bruising it, that gets swollen and then it starts to bleed. So that's kind of how vitamin C is linked up to collagen. And it has to deal with vitamin C is necessary for that post-translational modification of the proline and the lysine residues. Okay, so let's look at collagen in our bodies. So we can start with our skin first. Collagen can form, like I said, those fibrils. So we'll start here on the right side of this figure. So here, this is our triple helix. And remember, this is our left-handed triple helix here. And those helices all stack together and come together, and they link up covalently through that shift base formation from the lysine modifications, right, between those triple helices. So they form those fibers. Those fibers come together and form larger units called fibrils. And in our skin, those fibrils are more of a mesh-like network. Okay, that allows a kind of structural backbone for all the other cells in your skin and the different layers of your epidermis to grow and maintain the youthfulness of our skin. This is a short video done by the American Chemical Society, and it talks a little bit about skin getting old, anti-wrinkle, turn back the clock. And so some of the chemistry behind some of the things you might find in facial lotions or beauty creams that, people pay thousands of dollars for over their lifetime, so they just look younger. So just some more for your information fun videos to look at how that relates back to collagen. Okay, tendons. So again, almost from here. So tendons are long, those long, skinny fibers that are going to connect our muscles to our bones. And here again, you got to think the triple helix of collagen builds up these fibers. The fibers build up larger pieces called fibrils, and that's kind of what these shapes are. So those tendons are going to attach the muscle to the bone, and they're made up of solely collagen. Teeth and bone are going to be the last two we look at, and there's a lot of similarities between the two and some differences. So first off, we'll start with teeth. So this is an image of your tooth, right? You've got some enamel, that hard enamel on the outside, and some dentin. And dentin is made up of these tiny fluid-filled tubules through which your sensations are filled. So here's our gum, right? The gum line. All right, we're looking right here in this box. So here's our gum. This is our dentin, and then the enamel is going to be on the outside of our tooth. So I don't know if you have receding gums. That's where your gums start to recede back, and it exposes more of your dentin, and then you usually become more sensitive to hot or cold things. And that's because the hot or cold things that you're touching is affecting the fluid in these tubes by either expanding it or contracting it, and that somehow will signal to the nerve endings that, hey, something doesn't feel right, right? Like there is a change going on here and it's not what we want. But so let's talk about how this dentin structure is made or looks like, and then how collagen plays a role in that. So here is just an overall picture. Here are those tubules that are filled with fluid. And then what you'll see is here are those collagen fibers. So there's a bunch of collagen. It's kind of a mesh-like network, but it's more like rebarb. If you've ever seen any kind of concrete being poured, or you've heard of like a steel reinforced concrete, usually it's those wrought iron bars, right, that are there. And then you have you pour the concrete on top of it. So when I think of like the dentin structure, I feel like the collagen here is more like rebar because they're straight lines. Unlike the skin, which the skin has more of kind of flexible, squishy squishy collagen. Remember, there are 28 different types of collagen expressed in your body. So the primary sequence affects what the tertiary sequence looks like and affects overall kind of what the overall sequence is, the overall structure could end up looking like. The difference, though, also between the collagen found in your skin and the collagen found in your bones or in your teeth is that it's mineralized collagen. So there are minerals. Well, let's just take a quicker look. So we zoom in a little further. Okay, so now if we look at, these are those collagen fibers. And you can see, so here's kind of one of these circles represents a triple helix. So there's our triple helices. And again, the collagen molecules are only about 1,000-ish amino acids in length, maybe up to 1,400. So that's not the whole length of the tube. So what happens is they'll piece together. So they hold together covalently, but when you have teeth or bone, you also ossify or mineralize those collagen fibers together. So these black shapes represent the mineralization of the collagen. So right between where those two collagens might come together, there is a bunch of hydroxyapatite, which is calcium phosphate. Okay, so here's just kind of a picture of what that calcium phosphate structure might look like how those ions are all packed and bound and held together between those collagen fibers. Okay, so the problem is that over time you can eat some acidic foods, all right, and those acids can affect this crystal structure. So notice in the crystal structure here, it's 10 calciums, about 6 phosphates, and 2 hydroxides. Out of calcium, phosphate, and hydroxide, which one will readily react with an acid from maybe juices or you know, things? It's going to be that hydroxide, right? So hydroxide is going to react with the acid and protonate it, and you make a water molecule. So then that water molecule will leave, and that starts to weaken this crystal structure. So your crystal structure over time slowly starts to dissolve away due to those acids because you're removing those hydroxide ions. Okay, how can you fix it? I need to be able to fix it. So if we look, here's another kind of depiction of, here are these red dots are the calcium molecules. Yep, and then the phosphates are also kind of around in space, but in between the center, there's those hydroxide ions. Okay, so the acids react with the hydroxide and that's removed, and that weakens the stability of that crystal, right? And it dissolves. So fluoride toothpaste, though, works great. So fluoride is an ion. So this is a negative ion, the hydroxide, right? But fluoride is also a negative ion. I think it's right here on the periodic table above my head. Actually, no, is that the wrong side? It's all backwards. Yeah, that's beryllium. Sorry, fluoride's over here. But anyway, so fluoride can be a net negative 1 charge. So the fluoride in your toothpaste, what you're trying to do is you're trying to help rebuild the mineralization that's in between all those collagen fibers. And structurally, the fluoride is small enough to kind of take up the hole that that hydroxide is gone from. And then this is just another picture that kind of shows almost like how chaotic it looks. But you can see here are the phosphates, right? Here is that 3, right? The 3 calcium molecules with the fluoride molecule in green. So again, you're trying to rebuild that mineralization to hold those collagen in place. Okay, and that's pretty much what this is showing as well. So biofilms can be the films that are produced from bacteria. So if you don't brush your teeth, you get that plaque. That also is just the biofilm formation of a bunch of bacteria living in that area. And the biofilms can produce those hydrides, the acid, excrete acid into the environment. So not only is drinking like orange juice and stuff that's highly acidic, not good for your teeth, but eating lots of sugar is, because sugar feed the bacteria, and the bacteria produce hydroxide ions, and that it's doing the same thing that we just mentioned. So the hydroxide ions get in here and they neutralize the H plus. I am blanking. I'm so sorry. This is our hydroxide ions, and this is, well, hydronium. Okay, so those hydronium ions, or the H+ ions, come in and they react and neutralize our hydroxides. And they also can react with the phosphates, right? Because you can build that back up to look like a phosphoric acid, which still, again, destabilizes that crystalline structure because you're removing charges, like net charges. So the calcium is no longer getting to interact with as many negatively charged things as it had to hold that structure together. Okay, but again, fluoride is how you can try to combat that. So you can try to rebuild that crystalline structure with fluoride instead of the hydroxide. Okay, you can't brush your teeth with hydroxide. That would be terrible, right? That's a strong base. This is the last thing you would want to see in your toothpaste is hydroxide, right? Hydroxide is essentially what Drano is. It eats your hair that's clogging the drain. So fluoride's a safer alternative. Okay, lastly, bone. Bone and teeth are pretty similar as far as how it's kind of working and mineralizing is concerned. So if we look at, we've got bone here, we can kind of follow along. It's just showing different types of bone. That's probably, if you're really interested in what osteopathy, maybe you read the slide a lot more, but eventually here we go. We get down to that collagen fiber. The fibers made up of those fibrils, which here's our blown up scene of those. So here are our triple helices. Each one of these rows is our triple helix. And then these like shapes, these blobs are the bone crystal. Okay. So here is that bone, here is the structure of the bone with the collagen and the different crystals. So it's kind of a staggering pattern. And I liked this image because it kind of, again, tries to show you how potentially in that three-dimensional space over here, the calcium and the phosphates are coordinating and forming that hard salt-like crystal. Okay. So that pretty much sums up collagen and how it works in our bodies in different capacities. So, here's where you can reflect back and I can ask, what specifically about the structure of collagen aids in its function? So in order to form those fibers and be really strong, it's not normal alpha helices, it's that elongated kind of triple helix. So you need glycine residues, which are really flexible, to be able to kind of stretch and tweak those angles, those torsion angles, the phi and psi angles, to get that longer kind of helix, right? So that's primary structure for sure affects the overall structure of how collagen has it. It has many prolines in it because not only does it need to be slightly flexible so it can form a really elongated helix, but then it needs that rigidity to hold that helix. So that's why you see that toss between the most flexible amino acid and the most rigid or the most geometrically constrained amino acid, proline. Okay. The triple helix, so then you can talk, maybe the triple helix contains other amino acids that can become post-translationally modified. The lysines especially can become modified and ultimately cross-linked together so that multiple triple helices can come together in a covalent fashion that increases the strength there. And then, then in general, like collagen in your skin is more of a loosey-goosey mesh-like network. Collagen in your bones and in your teeth is mineralized. And collagen that makes up tendons is that like really long, strong rope-like structure. So so I'm sure you can think of more reasons and come up with some ideas yourself of how the structure of collagen aids in its function, but that there concludes my structure function analysis of collagen.